Okay, so we've gone through a breakdown of what all the objectives for this unit are. The next page, we're going to put our Unit 5 key vocab. And you should have this stuck in your notebook like this. I know this is a little different because typically I make you write all the words down, but there are a bunch of words for the sake of being pretty quick. I gave them to you. You'll notice I did leave a little space at the bottom because, as always, we always know that there's always going to be something comes up or a vocab word that we didn't remember or we didn't know about, so we can put a little space here that we can put stuff in. So I'm going to start off by going through some of these words. Obviously, not every vocab word that's here is going to make 100% sense to start off with. But as we go through the unit and we use these words and we become more comfortable with them, they're all going to make a lot more sense. But there's a few here that we can already start going through. So we know, don't we? We've heard, because we have talked about DNA plenty of times. So what is DNA? Well, we know it's the thing that carries our genetic code. We know already that it's a nucleic acid, right? It is very important that you know that DNA stands for deoxyribonucleic acid. Okay? And you can see there's the D, there's the N, and there's the A. Believe it or not, I've seen a question where they have actually referred to it as deoxyribonucleic acid, and people did not know that that's what they were talking about DNA, and started getting kind of like a little panicky because they'd never heard of deoxyribonucleic acid before. So I am determined that we are not going to make that mistake a second time. Okay, one thing I do want to point out is DNA starts with D. And the D stands for the sugar that makes up deoxyribose, ribonucleic acid, which is I could spell it right. Ribose. Okay, O-S-E. You remember O-S-E always tells me it's a sugar. So deoxyribose is the deoxyribo, the D part of DNA. Okay, and we'll get on to where that fits into everything here in a minute. Now, we have not talked very much about RNA. Okay, we know it's a nucleic acid, and RNA stands for ribonucleic acid. Okay. And obviously, there's the R, there's the N, and there's the A, okay? Now, what is the difference between DNA and RNA? Okay, well, one difference is in the sugar that makes up its building block, a nucleotide, whereas the D stands for deoxyribose sugar in RNA, the R in RNA stands for ribose. Okay, so it's a sugar because it's got OSE, but the sugar in RNA is ribose, the sugar in DNA is deoxyribose. Okay, all right, so what else do we know about these? All right, well, when we started doing biomolecules and nucleic acids right at the beginning of the school year, we talked about how DNA is a double helix, okay? And that's how one of the ways you know it, you're talking about DNA is because it is a double helix. Helix just means spiral, okay? So, and that would be DNA, okay? And RNA, kind of everything about RNA is the same, except it is a single helix. All right, it's only kind of got one side of nucleotides, as opposed to having two sides of nucleotides held together by hydrogen bonds. Okay, both DNA and RNA, we know both of them are made up from the same type of building block or monomer, and it's called a nucleotide. So this, right, this is the building block of nucleic acids. I'm sorry, I kind of squeezed these a little too close together, but you do already know this because you see it every single day on the wall in my room, right? You see that this is a nucleotide, okay? And these nucleotides are made up of three parts. We have the P for the phosphate, we have the S for the sugar, and we have the N for the nitrogen base. More about that later. But what is key here is if in DNA, right, we already talked about how the sugar is deoxyribose, right? So in DNA, that sugar is deoxyribose, okay? But in RNA, the only thing that's different is the sugar here is made up of not deoxyribose, but ribose sugar, okay? Now the nitrogen bases, we've talked about them. I'm going to talk about them here in a minute, but in DNA, we got ATGC, all right? In RNA, we have A, U, G, and C. So there's another difference there. DNA, ATGC, RNA, AUGC. Okay, and that we'll come to that here in a minute when we talk about complementary base pairing. Okay, it's kind of exactly the same with just a little twist to it. Okay, what is protein synthesis? Well, we know synthesis to make things. Okay, so basically protein synthesis is making proteins. And here's a neat bit. Making proteins. When you make proteins, you use the code in DNA, the genetic code, to determine what type of protein you're going to make. Well, since we're making proteins, right, I guess we need to know what amino acids are. Again, this is something we've already kind of covered. What are amino acids? They are the building blocks or the monomers of proteins. And okay, so here's a quick analogy. There are 20 amino acids. So since we're talking about building blocks, remember we talked about how a brick wall was made up of bricks, and like the entire wall is lots and lots and lots of bricks bonded together. Okay, imagine you've got a brick wall, but you've got 20 different colors of brick, right? And you can use any brick you want, any color. You can put all the same colors together. You can put different colors together. You can completely mix them up. That's pretty much a protein. A protein is made up from 20 different building blocks. They're all blocks. They're all amino acids. But the order of what color, what type of brick you're going to use is controlled by a set of instructions which are in your DNA. And I can't make protein synthesis any easier than that. It's basically putting a brick wall together with 20 different colored bricks, and you've got a set of instructions. So you've got to make sure you get those bricks in the right order. Okay, here's another word that we have used over and over again. We talk about genes. What are genes? again, keeping this as simple as possible. The ATGCs is your genetic code. One gene is 1, 2, 3 bases. So A, T, C, all right? Those 3 bases in that order would be a gene. So a gene is 3 bases on DNA. Okay. Now, a codon, this is where we kind of start getting into new territory, but a codon is similar to a gene, except it's 3 bases on a type of RNA called messenger RNA. And I guess at this point I should go back and tell you that RNA comes in 3 varieties, okay? We have mRNA, which is called messenger RNA. We have tRNA, which is called transfer RNA. Okay, those are 2 that we need to know about. And then there's rRNA, which is called ribosomal RNA, and that's, we don't really need to worry too much about that, so I'm not really going to talk about that. But if I have a type of RNA called messenger RNA, all right, mRNA, then that's 3 bases on the strand of RNA. So UAG would be what that would be, and that 3 bases on that would be RNA, codon. Okay, transcription and translation. These are 2 new terms. When we talked about in our objective about the process of making proteins, how we make proteins, it's split into 2 stages. Stage 1 is transcription. My entire life that I have been teaching biology and being a student, I have always made a mistake and struggled to remember which one comes in which order, and I eventually just came down to think, put them in alphabetical order. Transcription comes before translation. Transcription comes first, okay? And transcription is nothing more than taking DNA and turning it into a strand of messenger RNA, okay? And then translation, the second stage, is nothing more than using that strand of messenger RNA and turning it into a protein. Okay, it's that simple. Okay, when we did cells and we did the organelles and the things, we had ribosomes. They were the little tiny black dots that typically are stuck to that series of membranes called the endoplasmic reticulum, which is right next to the nucleus. And it's right next to the nucleus for a reason. Okay, but ribosomes, they are the site of protein synthesis. Ribosomes are a protein making factories. Okay? All right. A recap from when we talked about semi-conservative DNA replication and we talked about semi-conservative base pairing. So, in DNA, we know that, I'm sticking to my same color, A goes with orange, A goes with T, right? We know that. That's not very orange. Make that a little oranger. A goes with T, and we know that G goes with C. Okay, you'll remember that? I know you remember that because that's the one that we made 100 on the Unit 4 test where everybody was able to correctly complementary base pair one side of a DNA strand with the other. So we're good with that. A goes with T, G goes with C. In RNA, it's almost the same, except RNA does not have T. Instead of T, we have, my pens are drying out, we have a U. So A goes with U and G goes with C. Exactly the same. You said instead of an A, instead of a U, we have a T. Okay, next, and just to finish up here, okay, so messenger RNA, I'm not going to go into great detail on this. When we actually get on to the whole bit about how we make proteins, messenger RNA is mRNA, transfer RNA is tRNA, and these are two types of RNA, and they've each got a job. Obviously, messenger RNA is carrying some type of message, right? Transfer RNA, that's when that message gets transferred to proteins, and we'll go into these ones in a whole lot more detail. Okay, and that finally leaves us with this one, mutation. A lot of people get really confused and worked up about mutations. Mutations are not difficult. We talked a little bit in the breakdown about how they are, sometimes they can be neutral, they have no effect, and sometimes they can be bad, and sometimes they can be good. But basically, what do we need to know in terms of a mutation? It is nothing more than a change in the sequence, okay, of nitrogen bases. Okay, it's a change in a sequence of the nitrogen bases in a DNA strand. Okay, nitrogen bases in DNA. And since we know that the order of those ATGCs ultimately controls the protein that you produce, right? If you change the ATGCs, you end up with a different protein, and that is a mutation. Okay, those are your vocab. I know there's a lot to take in there, and this goes in your notebook, and then there is a little activity for you to do to help you learn these in Google Classroom.